Okay, good afternoon, morning to you all. I'm not sure what you, when you're viewing this, obviously, but this is the recorded portion of the class set 2 PowerPoints. Before we proceed, I just want to remind you all, please, that you have your first test on Monday. And that will be at the start of class, and it will be for half an hour. Out of 30 points. And there is a very important section that I want you to study in particular, which we will cover today, which you are definitely going to be asked on, and it's going to be a written question. Okay, so pay close attention. Okay, so this is where we ended up in class. We've discussed all the different kinds of cell forms and the different cell arrangements. And you will, I think by this time, have seen some of this in lab as well. And so we'll proceed from there. And we're going to talk about almost the most important feature of the cell, first of all, and that is the cell membrane. I have mentioned this before, and the importance of the cell membrane is that it allows the cell to have different conditions inside the membrane to those outside. In particular, cell membrane allows the cell to concentrate materials inside the cell, and that may be rare on the outside. In concentrating it, the cell facilitates biochemical reactions. So this cell membrane around the cell is the most important control mechanism for some simple and some very complicated reasons. So the cell membrane is made up of molecules which are called phospholipids. Now, a lipid is a fat or an oil. And here you can see here, this long chain here, those are lipid molecules. But they're not just lipid on their own. Instead, they have attached to them a phosphate group. And it's in fact, can be very complex. For the moment, just concentrate on this here. There's the phosphate group, and it's joined with a molecule called glycerol. So it's quite a complex structure, and it has a complex function because this portion is hydrophobic, typical of the oils. It does not dissolve in water. It is repelled by, and it repels water. That's why it's called hydrophobic, hates water. But the head here, this whole part here, is hydrophilic. That is, it loves water and it dissolves very, very easily in water. If I take a whole lot of these molecules and I mush them together in an aqueous environment, there is only one form that they can adopt which satisfies all of those properties. Remember, the tails need to be away from water entirely. The heads need to be dissolved in water because they're hydrophilic. Here's how they organize themselves. Here are the heads, here, dissolved in water on the outside. Here are the tails pointing away from the water. These are the hydrophobics. But the only way it could keep entirely away from water is to have another layer. Here's another layer, hydrophobic. Now there's a sandwich, hydrophobic, totally excluding water. Both sides, there and there, are dissolved in water, and everybody is happy. All of those molecules now are happy. And this is the very typical membrane, basic membrane structure, which is a lipid bilayer. That's what this is referred to as, a lipid bilayer. And we'll see this skeleton structure in all the membranes that we talk about. Now, in the bacteria, of course, we only have the cell membrane or plasma membrane around the outside of the cell. We don't have, excepting in some very special circumstances, we don't have membrane on the inside the way we do in eukaryote cells. We mentioned that already. So the really important thing about this lipid bilayer is because of its schizophrenic nature, the head's hydrophilic, the tail's hydrophobic. There are not many molecules which can pass easily across. And it excludes molecules like sugars and all sorts of other things that cannot make their way across through this lipid bilayer, thin as it is, just 2 molecules thick, and yet it acts as a semi-permeable membrane. So that means it only allows certain molecules to easily pass across. Things like gases that are dissolved in water, they can pass across very easily. So they pass across by simple processes of diffusion. That is, they move from areas of high concentration to areas of low concentration, and they can make their way across either direction. They can make their way across the membrane. So, for example, oxygen can permeate quite easily, can pass across. Carbon dioxide created in the cell, that can pass out quite easily. But many other things, including water, which can pass across a little bit, but not very much, they are selectively excluded from movement across. This gives the key to the real enormous function of the cell membrane. And that is embedded in that skeleton bilayer. There are many, many different proteins. Here you can see them here. If they are all the way across, then they're called integral proteins. That's it. They go from there to there. Some of them are just associated with one side or the other. But these proteins that are embedded in the cell membrane give the cell membrane its living nature. And that is because these proteins have many, many, many different functions. In the bacteria, we'll find that many physiological functions which depend on the presence of enzymes, proteinaceous enzymes, and other proteins, they all occur at the cell membrane. So, for example, the processes of respiration, of cellular respiration that we'll discuss in some detail, all of those proteins are associated with the cell membrane. But the other thing that happens is that these proteins control the passage of molecules across the membrane, which cannot otherwise easily pass. So many of these proteins are designed as gateways to allow molecules in the correct direction and to allow molecules to enter and accumulate in the cytoplasm, or waste products, etc., or enzymes that need to be released. Those can also pass through protein channels. So the cell membrane becomes involved with all of these different physiological functions, which is cell wall synthesis, we'll talk about in a minute. Energy metabolism, I've already talked about cellular respiration, but it's also the processes of photosynthesis, for example, are tied to the membrane. There is an involvement of the cell membrane in DNA replication. In fact, the circular chromosome that we talked about before is actually attached to the cell membrane, and replication of the DNA during cell division, that starts there where it's attached. There are many of these proteins which respond to different stimuli. Those stimuli can be the temperature, for example. They can respond to the presence of certain molecules, certain signal molecules. They can also respond to adverse conditions and result in the cell moving away from the adverse conditions, or they can sense the presence of favorable conditions and move the cell towards those. Don't forget, this little cell has no eyes and it has no ears. But these proteins that are embedded in the cell membrane are its eyes and ears, and continually sense what is happening in the environment and trigger responses in the cell. I've already emphasized the most one of the most important functions of these proteins is in transporting molecules. And they do so very, very selectively. First of all, there are a set of proteins which allow molecules, specific molecules, to move by diffusion. That is from areas of high concentration to low concentration. That is a passive process. It requires no input of energy by the cell. But many of these proteins are involved in active transport. And the reason that is so important is because it allows the cell to concentrate molecules inside, even when they're relatively rare outside. In that, if molecules are accumulated inside at high concentration and they are low outside, the process of diffusion would tend to force those molecules to move out. But the proteins act as gateways, and they grab hold of rare molecules out here, move them in, accumulate them here, and don't allow them to leave unless the cell needs to have them leave. Okay, so that's the cell membrane. We'll have reference to the cell membrane quite often during our discussion, especially about physiology. That outside the cell membrane, there is a cell wall. Now, I need to explain one thing to you about the cell membrane. I said to you that water molecules can sometimes make their way across. They don't do so very efficiently. But one set of proteins embedded in the membrane are specifically responsible for allowing the passage of water. And these are called the aquaporins. The name tells you what they do. Aquaporin, it means a pore for water. And they allow water to freely move across the membrane. The tendency is almost always for water to move from the outside to the inside of the cell. And the reason for that is that this is such a rich solution that water is relatively rare inside the cell, but it's abundant in the surrounding environment. So water tends to continuously move into the interior of cells. This is something that happens not only with bacteria, it happens with us, it happens with fish, it happens with plants. Always the tendency is for water from the environment to move into the interior of the cell by diffusion through aquaporins. That's fine. That means the cell's always got an abundant source of water, which it needs all the time. But it creates one problem. And that is this cell membrane, there is very little tensile strength to the membrane. And if we have water flooding into the cell the whole time, the cell starts to swell. And this weak cell membrane bursts very, very easily if you allow the cell to swell. This is the principal function of the cell wall around the outside of the cell. In bacteria, it has some specific properties that I'll mention in a minute. But even plant cells, for example, have a cell wall, and that for much the same reason. We have other mechanisms to cope with the inflooding of water. What happens is this cell wall around the outside, first of all, it gives the cell its shape. So its shape is genetically determined. It's built by the cell membrane, but its shape is genetically determined. It's rigid, it's very, very strong, and it has no selective permeability. It's rather like a mesh, like a net. So molecules can pass freely through it without impediment. The reason it's there is so that when the cell swells because of the diffusion of water is called osmosis. When the cell swells because of osmosis, pouring water into the interior of the cell swells against the inside of the cell wall. and that stops the cell from bursting. So the primary feature of the cell wall is to prevent the bursting or lysis of a cell because of osmotic stress. So that's the main reason. It does offer some protection probably from predators or whatever, but its main function is as an osmotic control. There are, however, some features of cell walls which are of clinical significance. And this is particularly in one type of cell wall that we'll talk about in a second. The gram-positive cell wall excites strong immune responses from the host if this is a parasitic pathogenic bacteria. But the structure of the cell wall, the building of the cell wall, is subject to control by antibiotics. So this is the main function of the whole group of antibiotics associated with the penicillins, ampicillin, amoxicillin, all of those cillins. They all affect the structuring of the cell wall, the building of the cell wall. And when they do that, they weaken the cell wall and the cells burst due to osmotic stress. In natural systems, in us, for instance, there's a very common enzyme which is produced in body fluids. We have it in tears and mucous from mucous membranes, etc. And it is called lysozyme. And lysozyme actually breaks down the cell walls of bacteria. And so it's there as a disinfectant, really, in our tears and other body fluids. Okay, so let's have a look at the actual molecular structure. And don't panic, we do it really simply. The first thing you need to know is that the principal component of the cell wall, the structuring of the cell wall, is by this molecule called peptidoglycan, a very strong molecule that is made up of sugar units. You don't need to remember the two names. There are two sugar units, ding, 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 which get joined together to form these rods. And those rods, the initial joining is by a group of proteins here, which link them all together. So they're linked by proteins into sheets like that. Now, those sheets, there are many of them sometimes that are joined together. And they are joined together by molecules called teichoic acid. So here is the first type of cell wall that we're going to discuss. And this is the gram-positive cell wall. I think I've got a bit of control. So the gram-positive cell wall consists of many of those peptidoglycan sheets, and they are anchored together by, for example, by this molecule here, teichoic acid, which actually not only links them, it ties the sheets to the cell membrane. Then there are other proteins which link all of these sheets together into a very, very strong meshwork that forms the cell wall. So here's the thing to note about the gram-positive cell wall. First of all, it is a thick layer of peptidoglycan that is tied to the cell membrane by teichoic acid. And all of the peptidoglycan rods that you see here are all tied together with protein. Take careful note of this, because on Monday in your test, you are going to be asked to compare this gram-positive cell wall structure to the gram-negative cell wall, which we're going to talk about in a minute. There is a clinical significance to these teichoic acid molecules here. And teichoic acid will actually spark a very powerful immune response if it is liberated into the bloodstream. So it's whenever we have circulating bacteria which we might want to get rid of from body fluids, you have to do it with antibiotics very, very carefully, because you don't want the cells just bursting and liberating all of these molecules free into the blood circulatory system. And so in any systemic infection, antibiotic treatment needs to be very carefully controlled. So this is the gram-positive. Let me just see if I've covered everything. Yes. So this is what happens with penicillin. Penicillin stops all of these linkages being built up, or interferes with all of these linkages on the peptidoglycan. And so the peptidoglycan sheets simply don't form, or they break down. And once that happens, the cell loses osmotic protection and the cell bursts from osmotic stress. That's the gram-positive. Things like bacillus, all of the different bacilli, all the rods with these thick cell walls, those are gram-positive. The other kind of cell wall is a much more complicated structure, and this is the gram-negative. You might think by the name gram-negative that it actually would be less complex. It's not. It's much more complex, but we'll start right here. This is what is identical to the gram-positive. It also, of course, has this cell membrane there. On the outside of the cell membrane, however, it only has a very thin layer of loosely bound peptidoglycan, very thin. And on the outside of the peptidoglycan layer, there is another membrane structure. This is called the outer membrane. This whole structure here is the outer membrane. It is built on the same principles as the gram-positive, as the cell membrane. And that is, we have hydrophilic heads and hydrophobic tails forming a bilayer. But only this layer here is really very similar to the normal lipid layer, phospholipid layer. This layer on the outside is different. It has embedded in very rich concentration of this molecule here. I'll show you another picture in a second. And this is not a phospholipid, but it is a lipopolysaccharide lipid there, and then a head here with many very complex polysaccharide sugar groups attached to the head. So this is called a lipopolysaccharide. It's lipid there with polysaccharides attached. And this is very significant clinically because this lipopolysaccharide excites a very powerful immune response, a very, very powerful immune response in a host. And as a result, its liberation into the bloodstream can be absolutely catastrophic. Gram-negative bacteria are notorious, absolutely notorious, for causing a condition called sepsis. And sepsis is an immune response. It is an overwhelming immune response globally in the body, which is a clinically critical situation. It can lead very easily and quickly lead to death. Sorry, I just had to stop for a minute. There's a huge thundercloud passing overhead and the whole room went pitch black. Now I'll switch some lights on. You may hear some thunder in a minute, I think. Okay, so here are the basic features of the gram-negative cell wall. We've got a cell membrane, as always. We've got a thin layer of peptidoglycan. Oh, And then on the outside of the peptidoglycan, there is an outer membrane. And the outer membrane consists of a phospholipid layer, a phospholipid skeleton in this leaflet here, in the outer leaflet. But in that outer leaflet, there are many of these lipopolysaccharides. These are like phospholipids with the polysaccharide groups attached to them. So here is the lipopolysaccharide structure. And you can see here, this is called lipid A. The lipid part is called lipid A. And lipid A is what results in this the clinical condition, septic shock or sepsis. And that is, as I told you, is a life-threatening condition. Then on the outside here are polysaccharides. There are many different versions of this lipopolysaccharide molecule. So there are many of them have different polysaccharides attached to them. It depends on the species of bacteria. And but it's this lipid A. It functions as an endotoxin. An endotoxin is something which is inherently part of bacterial structure, which when it is liberated, acts as a toxin in the host. And in case of gram-negative bacteria, clinical treatment has to be done extremely judiciously. You cannot simply flood a person with antibiotics, kill off all of the bacteria, have them first disintegrate, and release lipid A into circulation, because it will result in a very, very severe immune response from the host. I'm sorry. I just want to go back here. There's just one or two other things that I need to tell you. If we got this outer membrane here and our cell membrane there, in between them, there is a space. And that is called the periplasmic space. It is not filled with cytoplasm. It is filled with what is known as periplasm. So the cytoplasm is on the inside. Periplasm fills the periplasmic space. This has an important function because also embedded in the outer membrane, just as we would have had in the cell membrane, are many, many proteins. And many of the proteins of the outer membrane are concerned with capturing and transporting relatively rare molecules from the environment and accumulating them in the periplasm. Gram-negative bacteria are very, very good at surviving, for example, in poor nutrient conditions. And we often find, like in marine systems and in freshwater systems, there are very few gram-positive, free-living gram-positives. Most of the free-living bacteria are gram-negative for that very reason. They survive much better in low nutrient conditions. So we've got cell membrane, outer membrane, periplasmic space here, and only a thin layer of peptidoglycan. I'm not going to go into all the details, but I will just point out to you that this is the reason why one of our most important clinical tests is to discern whether bacteria are gram-positive or gram-negative, and this is how it works. In a gram-positive bacterium, the staining, it reacts differently because of this thick layer here of peptidoglycan. We get this peptidoglycan layer results ultimately in the cell staining deep blue-black with the gram stain. In the gram-negative does not stain blue-black. It doesn't, the stain does not stick in because to the cell because it doesn't have a thick peptidoglycan layer. And we end up in the gram stain with the gram-negative staining pink. So that will be discussed in more detail when you get to the lab. Okay, so there's one caveat to all of this, and that is there is some variability in the structure of cell walls for different bacteria. And this is one group which does not respond to the Gram stain reactions. So we can't say whether it can't tell whether it's Gram positive or Gram negative. And these are an important group, clinical group of bacteria called the mycobacteria. They cause, for example, they cause tuberculosis, Mycobacterium tuberculosis, and they cause leprosy, Mycobacterium leprae. There are others which are harmless, but they will show the same cell wall response. And the reason for that really are Gram positives, they really are Gram positives, but included in that cell wall is a lot of this molecule called mycolic acid, and it's a wax. And because it's a wax, and all of our Gram reactions are based on aqueous dyes, it repels the dyes, and it will not stain. You have to use a special stain technique called the acid-fast stain procedure. And this you will hear about again in lab. But the end point of an acid-fast stain procedure is that any bacteria which have mycolic acid end up staining this color. Here they are here. And these are actually bacteria of tuberculosis, Mycobacterium tuberculosis, long rods. And you can see they stain a distinctive red color. Okay, so I'm just checking my time. So I hinted before at the fact that the proteins embedded in the cell membrane are responsible for many of the functions of the cell, but especially the control of passage of molecules across the membrane, across the semi-permeable cell membrane. Most molecules will not pass easily across that cell membrane. Gases do. And here, for example, I don't know, oxygen or whatever, on the outside, they will pass across the cell membrane due to simple diffusion. So diffusion occurs because there is a high concentration on one side and a low concentration on the other side of the membrane. The tendency of molecules to be under those conditions is to move from an area of high concentration to an area of low concentration. And that is simple diffusion. For most molecules are going to attempt or be driven to undertake that movement, but most molecules can't pass across the membrane. So they don't make their way to the inside. Gases, things like that, they can. In order to get molecules to pass across, they have to interact with proteins which are embedded in the membrane called the transport proteins. And there are two very important distinctions in the functions. One set of proteins exploits the simple diffusion process. You can think of it this way. The proteins are often very specific to a molecule or a group of molecules. So when the cell needs molecules on the inside, the proteins open and the molecules pass across. And they pass across by simple diffusive processes. And this is called facilitated diffusion. The protein is facilitating the diffusion. It's exerting control, but when it allows passage of molecules, the molecules simply go down a concentration gradient from high to low. So that is facilitated diffusion. Some of them, there are some which are non-specific, which allow transport. But there are others which are, many of them are in fact extremely specific to a particular molecule. And this one here is the transporter for glucose. It only allows the transport of glucose across, and it does so by changing shape. It actually, tempting to think of it swallowing the glucose molecule and spitting it out on the other side. And that is facilitated diffusion. There is one specific group of proteins adopting this facilitated diffusion, and those are those which move water. And I've already given you the name of those. These are the aquaporins. And they are the ones which allow water to pass across the cell membrane. A little bit of water can make its way across, but it's very inefficient. Instead, the aquaporins allow the free passage of water to the inside of the cell. And they don't actually exert very much control over the passage of water. As long as there is an aquaporin, water will pass across. So one of the ways the cell can control the amount of water coming in is to control the number of aquaporins. And these can be manufactured and embedded or broken down and taken away. So there is some dynamic nature to the occurrence of the aquaporins. Okay, so let me just go back here. Notice something very important here, and that is that the cell is not exerting any energy to move these molecules across by diffusion. The energy comes from the concentration of molecules, forcing them to move from an area of high concentration to low concentration. The proteins do not, the cell does not use any energy to affect that movement. There is another set of proteins, tremendously important, embedded in membranes, which actually do. They actually require an input of energy in order to transport molecules. So here are all our passive diffusive processes, okay? So, and this here is an active transporter. This is a protein which is going to utilize the biological source of energy, which is adenosine triphosphate, or ATP, and it's going to use some energy to move molecules. And what is critical here is all of these diffusive processes here, they allow movement from high concentration to low concentration. Active transport allows the transport against a concentration gradient into an area of already high concentration. So these are very important critical molecules, excuse me, which are being moved against a concentration gradient. So we can think of, for example, molecules which are accumulated in the cytoplasm, which only occur in low concentration on the outside. But these proteins, these active transport proteins, will move them from low concentration to high concentration, and that requires an input of energy from ATP. That's why we call it active transport. Be prepared on Monday in your test to be able to look at this diagram here and explain to me what is happening. What is the difference between these different transport proteins? Okay, that's going to be another question on your test on Monday. Let me just check my time. Okay, we're fine. So that's the outside, okay, part of the outside. We will return to the outside. Just mention again some of the things I actually mentioned earlier in the week. But let's have a look at the inside of the cell, inside the cytoplasm of the bacterial cell, and see what we have occurring there. The first is that there is a large circular chromosome. That chromosome is not bound by a membrane. That is a feature of the eukaryotes. The prokaryotes do not have a membrane enclosed nucleus, nor any other membrane enclosed organelle. They may also have small circlets that you've heard about before, and these are the plasmids. Small circles of DNA. Those plasmids often contain very useful information. For example, they may contain information which confers resistance to antibiotics. That's one thing. They may contain information which allows a non-pathogenic cell to become pathogenic. that's the other. And so these little plasmids are extremely interesting, because if a cell dies, for example, and bursts in the environment, these little bits of circular DNA, they actually survive really well. And many bacteria are adapted and very good at bringing those into the cell from the environment. So bacteria can actually transfer information from one cell to another, and that information is often very useful to the cell that acquires it. We say that plasmids are infectious. They can move from one cell to another. Also included, as in all living things, are the ribosomes. And the ribosomes are responsible for synthesis of protein based on instructions from DNA. And in the bacteria, don't worry over much about the sizes and everything. I just want to mention one thing about the ribosomes, and that is bacteria have distinctive ribosomes. They have distinctive molecules in their ribosomes, which are slightly different to the eukaryotes. Please just read through these other inclusions. I'm not going to do them in detail. Now, here's another feature of the interior of the cell, which you're probably going to see in lab sometime. I believe we have a special staining technique we'll teach you for looking for these. Certain of the gram-positive rod bacteria, namely Clostridium and the genus Bacillus, are able to form spores. Now, the bacterial spore is very, very interesting. It's a strange thing. It is a specialized cell. Normally, you realize that bacterial cells will grow and then divide equally from two daughter cells. When Clostridium or Bacillus grow in adverse conditions, they begin to divide differently. Instead of dividing equally, they divide, here they duplicate the DNA, and they divide so that there is an unequal, there is a small cell. And that small cell gets invested with a very, very interesting cell wall. This cell wall is highly protective. It protects the interior, the cytoplasm on the interior, from radiation, from disinfectants, from heat. You can boil spores and they will survive. These are very, very resistant and very, very long-lived. They're also extremely small, so they can be blown around very, very easily. And when they land in suitable conditions, they germinate and they form a normal functional vegetative cell. They are extremely long-lived. And in fact, some have been recovered, for example, from tombs of the mummies, thousands and thousands of years old, and put into medium, they immediately germinate. So there's inside the cell, there's virtually no metabolism at all. It's in a resting state, protected by this thick cell wall. And this is called, these are referred to as endospores, because they form inside the mother cell. Okay. There are also features on the outside of the cell wall. And one of them that you will see in lab is referred to, the whole thing is referred to as the glycocalyx. But the glycocalyx may come in different guises. The most commonly seen is when it forms a very large conglomeration of these slimy mucus-like molecules, mucoid molecules that surround the cell. And this is called the capsule. And the capsule is partly protective. It protects against predators and things like that. It protects against, has a certain amount of protection against viruses. But its most important feature is that it allows the cell to attach. And attachment in bacteria is very important. It's often attachment triggers the cell to go into high metabolic mode and begin growing rapidly and then dividing and so on. So it's an important environmental property to have a capsule, but it's also very important in pathogens. And the attachment of cells is often the first step in establishing a pathogenic relationship with the host. So this is the capsule. You use a special staining technique to visualize it. Here are the cells on the inside, and this clear area marks where the capsule is. Here's an electron micrograph. You see this very loose molecules all the way around. If the molecules on the outside of the cell wall, if the glycocalyx is very loose, we call it a slime layer. But its composition is essentially the same as the capsule. It's just there are fewer molecules and they're much more loosely attached, often much longer too. All right, and so I also told you, I had mentioned this before, that the many bacteria move by nature of their flagella, which are attached. They're actually attached to the cell membrane, but they penetrate through the cell wall to the outside. And we can classify bacteria by the type and nature of their flagella. So here, for example, this is very common. This is a cell with flagella all over, coming from all regions of the cell. And this is called peritrichous flagellation. This is called monotrichous flagellation. There's just one, and it is polar, comes from the pole of the cell. This is called lophotrichous. Lophotrichous means a bunch. So there are a whole lot of flagella here, and they are also polar. This is amphitrichous. That means that at both ends. This is spirillum, which characteristically has very large flagella, one of the few bacterial flagella that you can see with regular staining. They have something specialized structure, but they are flagella. Normally, flagella are invisible. You have to use very special staining techniques to visualize them, or electron micrograph. This is amphitrichous, both ends, and both are polar. Okay. I mentioned this before. I'm not going to do it in any great detail now. The flagellum is embedded in the cell membrane, and it has a little tiny motor that is run by effectively by electricity, and spins the whole flagellum. It does not induce a wave. It causes the whole flagellum to spin. Fimbriae and pili I have talked about before. Pili especially are responsible for two other kinds of movement, and this is movement usually associated with a surface, and they are twitching and gliding. Twitching, it's got a strange jumping motion to it. Gliding, exactly what it says. It looks like the bacterium is cruising along the surface. And fimbriae are mostly associated with attachment, functioning together with the glycocalyx in order to attach bacteria. There's one specialized kind of pilus. Pilus, by the way, means arrow. And this is called a sex pilus. And these are in bacteria which can transfer DNA directly from one cell to another. They don't what happens is this pilus establishes a contact between one cell and another cell, and then the pilus contracts and it pulls this cell tightly against the other, and the pilus forms a pore in the cell membrane. And this cell then can pass DNA into this cell. It's always only one way. It passes from one cell to the other. This cell does not acquire from the other cell. Okay, so I'm going to stop there, and we will. I'll just cover this little section on eukaryote cells on Monday after your test. Please remember the two things that you know you are going to be asked about, and that is comparison of the gram-positive and gram-negative cell wall structures. And then you're going to be asked about the distinctions between the different kinds of transport proteins or different kinds of transport across the cell membrane. Okay, and I'll see you then on Monday.